So welcome. So excited to see everyone. I wish everyone could have this view. It's an amazing view to see so many of you interested in clinical trials. My name is Brad Monk, and I will serve as the moderator for this afternoon's symposium entitled Emerging Global Therapies and Clinical Trials in Progress for Endometrial and Ovarian Cancers, Understanding the Landscape and Optimizing Strategies. So I apologize to those of us that do cervical cancer research. We have other symposia for that. We thought we should pick two, and these are the ones that we picked. But here's the point. We are here at an international symposium, and we're going to hear about international trials by international experts. So this is so exciting. We hope that this is a platform for satellite symposia at upcoming IGCS meetings, because without research, we cannot cure more patients, which is our common goal. These are our beautiful faculty. I'm going to have each of them come up very soon to the podium and introduce themselves. I want to thank Jenna Cummins and her staff for all the beautiful work, and if that isn't a beautiful picture, I don't know what is, except for that silly guy over there on the side. So, I can't help that. We would like to thank our commercial supporters. Importantly, this is an independent symposium, so we will be fair, balanced, and objective. I want to thank Gilead, Karyopharm Therapeutics, Genmab, Immunogen, Corcept Therapeutics, and Sutro Biopharma. So if you could, and I know you're reading, I apologize, this is important. So if you grab your phones, I can see you whether you're doing it or not. If you could grab your phones and take this QR code, we're interested in sort of learning about your needs. We use this information for grant submission. So we want to know who you are, so when we go to sponsors, we can say this is what that person, you, and so on wants to know. Our goal is to deliver what you're interested in, and this survey will help you. You can write down that code, 3156053. All right, these are our objectives. We want to analyze, explore, assess, and understand. Those are all verbs. We want this to be an active engagement. There are key components in clinical trials and in the progress that we've made in the past. We want to explore the future. It's very competitive. But remember, we don't compete against each other. We compete together against the cancer. We want to assess the past body of knowledge and impact these trials, which are very important. And again, these are international trials. This is an international community, and this is an international fight. And ultimately understand the global footprint of these clinical trials as we progress. These are the disclosures. They're also on the website. All of these have been vetted. So we'll start here, and I'm doing some introductions here, but I want to hear about the international footprint. And I apologize, we couldn't have every region of the world. Actually, I think we do have every region of the world, but we don't have every group in the world. But we can't succeed alone. So we're going to go through here, and we're going to hear about the collaborations that we do together, okay? And so I'm going to have each of these kind individuals come up and introduce who they are, who their groups are, what they represent, and most importantly, how they help us cure cancer. Thank you, Professor Colombo.
Thank you. Thank you, Brad. Thank you. It is indeed my great pleasure to be here on behalf of ENGOT. I'm actually the immediate past chair of ENGOT. Up to 2 weeks ago, I was the chair. But it's very important for me to tell you about ENGOT because maybe not all of you are completely familiar with this organization. What is ENGOT? ENGOT is the European Network of Gynecological Oncological Trial Group. As you can see, covers almost all Europe, You can see here 21 cooperative groups included in ENGOT, and 33 countries, because some of the groups include more than one country. On the other hand, we have also countries with 2 groups. But the important thing is that all Europe is covered. How do we run a trial? We have several publications underlying the way we are working and how we work, particularly with companies, with industries, because it's so important to have a nice collaboration with them in order to develop new drugs and more options, more therapeutic options for our patients. So you may want to read these papers just to understand what we established as important collaboration. And in fact, we value particularly the collaboration with industry, because we do believe that it's crucial that industry listen to us, listen to the academic. We want to develop trials which are patient-oriented. We want to focus on unmet medical needs. And I think for industry, the academic participation and validation of clinical trials design is a plus for credibility. And also, we established a clinically oriented translational research design for almost all our trials. And it is indeed an opportunity of helping in the development of the pipeline from the beginning. So what we try to do with the industry at the very beginning, when a new drug is coming into the clinic, we want to participate starting from the phase 1, and then if it goes on in phase 2 and phase 3, so that we are really part of all the development of a new drug. But of course, the partnership with companies in the industry is important, but even more, as Brad said, we have to work together against the cancer. We are not competing each other. We want to collaborate. We are collaborating effectively. And we have this liaison committee with the GOG established a few years ago by Ignace Vergoot, and is working so well, because in this way we can really share our experience and know what's going on and try to lead the trials in a very fair manner. So there will be trials led by GOG with the participation of ENGOT and vice versa, and we try to keep the balance so that everyone will be happy, and of course, all women across the world will have access to the new and most important trials. These are some of the achievements, just to let you know, in these 18 years of the life of ENGOT. Here you see the number of trials we run according to different gynecological cancers, and altogether 152 trials. Well, the Model A, B, C is, Model A is mainly academic, and Model C is a sponsored trial. And you see the number of publications that we achieved. So finally, just to sum up, what is our mission? Our mission is to bring the best treatment to gynecological cancer patients through the best science, and enabling every patient in every European country to access a clinical trial. And partnership with ENGOT from the industry can lead to a wise development of strategies and pipelines of different companies by supporting all in a fair manner. Thank you.
Thank you. Thank you, Professor Colombo, for all the great lessons and all the great studies that ENGOT's doing. We're now going to transition to Asia. We've invited Professor Jae-hoon Kim from Korea to tell us about the KGOG. Thank you.
Hello, everyone. I'm happy to introduce the Korean Gynecology Oncology Group. You can see a world map with Korea highlighted in red. Korea is located on the northeast part of Asia and has a population of more than 50 million people. You can see the logo of KGOG. Somebody believed that the red portion is communism of North Korea, and the blue one is the democracy of South Korea, but it is totally not. It just means harmonization. Our mission and vision is to advance research and partnership in GYN oncology. We collaborate closely with researchers worldwide and are dedicated to supporting investigator or sponsor-initiated trials. We aim to strengthen partnership with organizations like GOG, sometimes ENGOT, to expand the studies and elevate treatment standards through rigorous clinical trials. KGOG was established in the fall of 2022 as an academic society. In 2025, we became a provisional member of GOG Legacy and joined the GCIG in 2027. In 2020, we were re-established as a nonprofit incorporated association. Last year, we published a 20-year history of our achievements. Today, KGOG includes more than 200 gynecology oncology specialists affiliated with 78 hospitals. KGOG protocols can be categorized. First, national granted projects. Second, investigator-initiated trials and sponsor-initiated trials. Third, KGOG-led study or participated study. And fourth, is global trials. Currently, we have conducted 49 clinical trials for cervical cancer, 35 for uterine cancer, 68 for ovarian cancer, and 12 for other cancers, including retrospective studies. Our global research primarily has collaborated with big groups, resulting in over 40 studies until now. KGOG offers key services to enhance clinical trials for sponsors, consultation and protocol design, site selection assistance. Our KGOG team guides the selection of well-equipped trial sites, financial management, and stakeholder communications. KGOG's monthly meeting promote collaboration and clear communication among sites, CROs, and sponsors. You can gain a better understanding of KGOG's everything by reading the recently published 20 years of KGOG. Thank you so much.
Thank you. So we're going to stay in Asia, one of my favorite parts of the world, by the way. We'll get to South America soon.
Sorry.
I love that. So we're going to now hear about a group that may be less familiar to you. It's called GOTJ, and we've asked their founder and president to speak to us, and also happens to be the president of our wonderful society. Thank you for all your work, Professor Fujiwara, not only for what you're going to talk about, for making this a successful meeting. Thank you.
Well, thank you, Bharat. This is a very kind introduction to me. Okay, so the GOTJ stands for the Global Oncology Trials Japan. So GOTJ is much easier to say, okay? So this is the structure of the GOTJ. So GOTJ has been known as the Energy Oncology Japan for years. And the Energy Oncology Japan was formerly a GOG Japan since 2002, when we started the collaboration with the NCI-sponsored trial with the GOG and NRG. So we have energy trials not open, not just for the GYN. We have the lung cancer group, breast cancer group, head and neck, and many, many other brain tumors too. So we have approximately 20 sites which has been approved as NCI-approved the trial sites. And in addition, we have started to collaborate with the partner CRO to help those academic trials, which has already know the expertise in the Japanese regulations. And we have conducted the registration-directed trials, which is energy and GOG trials get the approval inside Japan. So 2 years ago, we have suggested by the energy leadership that we need to expand our abilities to partner with to power up with the GOG partner trials too. So we renamed from the Energy Oncology Trials Japan to GOG-J. Now the GOG-J is a parent institution for the NCI, the energy the sites, as the energy sites. And also we have started to conduct the pharma company trials in collaboration with the GOG Foundation, GOG partners. So we have the master service agreement with GOG, and we have just started the new trials recently. So our strength is, we have, as I said, strong partnership with GOG Foundation, and we have the very good, positive communication with the regulatory authorities, which means Japanese authorities, as the academia, not as a supporter, not only for the support of the pharma company. We have the strong the relationship with the PMDA and the health ministries. And also we have the close collaboration with the sites that have previously conducted the clinical trials. So as I said, we have 20 NCI approved sites, and in addition, we have another 20 very strong sites to be able to conduct the pharma company trials. And also, as I said, we have the integrated partnership, the preferred and CRO. Now we are expanding our ability to have the relationship with the site management organization pretty soon. So, very ambitious. So I'd like to explain the meaning of the logo. Just looks like a tropical bird, very colorful. So the blue means global outlook, and red means a passion. We all has a passion, not a communism, okay? And the black is resilience and toughness, and green is sustainability. Thank you so much.
Thank you. All right, now we get to go to the Americas. So thank you. I'd like to introduce to you Graziella Dalmolin from Sao Paulo. Thank you for coming all this way, for agreeing to be with us and talk about a very large continent that's very important to us in doing clinical trials. Thank you and welcome.
Thank you so much. Good afternoon. I would like to thank again the invitation to be here, and it's an honor to present two important cooperative groups in Latin America: the Brazilian Gynecologic Oncology Group, EVA, and the Latin America Cooperative Oncology Group, LACOG. So EVA, different from the other cooperative groups that were presented here, EVA is not only a research group. Our mission is about education, research, and advocacy with the collaboration of different societies. We're now 715 members, including physicians from all areas in gynecology, oncology, including also nurses and pharmacists. And our international collaboration includes GOG, GCIG, and SGO. For advocacy, we have two important projects: one in cervical cancer with partnership with WHO and PAHO, and one important awareness of gynecological cancer month in September that has a partnership with IPVS. And for research, we do a partnership with LACOG and GOG, and now we have 8 ongoing studies, 2 that will be presented here, Rosella and Gloriosa. And here we have our collaboration with GOG and EVA. We're proud of our advocacy with the Cervical Cancer Elimination Day in Rio. And it's a a group that is increasing every year. We also have LACOG. Differently from EVA, LACOG is mainly a research group in Latin America with all types of solid tumors, and that's why we do a partnership with EVA. LACOG has now more than 620 investigators in 17 countries in Latin America. Now there's more than 200 hospitals and research sites, and more than 20,000 patients were enrolled since the beginning of LACOG. Here now we have 53 ongoing studies in 119 sites, now in 9 different countries in Latin America. Thank you.
That's great, thank you. Now we're going to go from South America to North America. I'd like to welcome Stephanie LaRue. Stephanie LaRue is a world-renowned investigator from the Princess Margaret in Toronto, Canada, and is going to speak to us about our Canadian sites and how important they are in our clinical trials. Thank you.
Thank you so much for having me. It's a pleasure to represent Canada here. In Canada, we currently have 2 cooperative groups. One is the Canadian Cancer Trial Groups, and I will say that in French as well because it's a bilingual country, the Groupe Canadien des Essais sur le Cancer. This group is actually focused on cancer, including a specific branch on gynecological cancer. And you also have the Princess Margaret Consortium that I will focus on today. And as you can see, we have in common the Canadian flag and the maple leaf, which represent well Canada. So here the Princess Margaret Consortium network. So it's important. Canada is a big country, and the health is based on provinces. So it's very important that we collaborate between our provinces. And we have different sites that have specific agreement with the Princess Margaret Consortium to launch very quickly different trials. So we go from the west to the east coast to ensure that we offer all patient clinical trial. So in terms of the Princess Margaret Consortium, what is our mission? Is to ensure that we have a trial network across Canada to offer options for all our patients. But it's also very important to collaborate internationally, so to collaborate with ENGOT and GOG to ensure that we have international collaboration for our patients as well. One of the other missions we have is really to develop a pipeline of treatment options for our patients at the different time point of the journey, including the prevention to the and at the time of platinum resistance. So it's very important to ensure we have options at the different time of the patient journey, that we answer different questions. And the last point that is very important for us is actually to try to incorporate robust translational research to try to understand the disease biology to move forward the improvement in the patient with ovarian cancer or gynecological cancer, because I think the only way to understand and improve outcome is to understand the disease biology. So it's very important to have this correlative analysis built as part of clinical trial and to identify specific biomarker to know why patients respond or not to the treatment to leverage the next step trial. So when you want to have a trial network, it's very important to have oncology expertise. So it's why it's very important to have a network that you can rely on. This network is actually the largest Canadian early phase trial academic program. In the last decade, we have enrolled more than 5,000 patients across different trials. And we have a robust infrastructure and expertise in a multiple sites. And we have identified PI at the different sites that we can collaborate with. And we really have an emphasis on specific investigator and translational research studies. And to be able to actually launch investigator-initiated trial is very important to be supported. You need to have the right infrastructure in place to conduct this specific investigator-initiated trial. So it's why we have actually a specific Canadian CRO that we work with closely to actually launch this specific ITT trial. And also what I really highlight, and again, as we say, it's very, if we want to go far, and as you can see later on, there's multiple clinical trials. So it's very important to collaborate, to include the patient very quickly on trial to answer the question appropriately. So collaboration internationally, and we also are part of the GOG network to launch this specific clinical trial. So thank you.
Yeah, that's impressive. And again, thank you all for being here. I want to show a few slides about the GOG Foundation. The GOG Foundation, which I'll show the timeline in a minute, is a nonprofit organization whereby it is the contractual entity for what we call GOG Partners and Energy Oncology. These sites are shared. GOG Partners has about 300 sites. Energy Oncology has over 100. We don't open all studies at every site. That's important, and that's part of what we do is we help you find the best site for your trial. We try to manage competition. We don't want sites to open competitive trials, and we try through the contractual business organization operationalize quickly. We do a lot of things other than clinical trials. We help design the trials and consult, and then we do what we're doing here. We educate, because no matter how many clinical trials you do and report, unless people learn about them, we have really not accomplished our goal. So our mission is to conduct the trial that positively impacts patients. Mostly we do therapeutic trials, but we have done some prevention, and we definitely have some surgical trials, as you know, including a HIPEC trial. And hence the name, we want to be the collaborative organization. Now, we've been at this a while. The gynecologic oncology group, which doesn't exist anymore, was started in 1970. So if someone says gynecologic oncology group, it doesn't exist. We incorporated to create this business entity, and then in 2010, co-founded with Tate Thigpen, GOG Partners. We started, and we really sort of modeled after what you had nicely explained, Nicoletta Colombo, what ENGOT was doing. So you started 4 years before us, and ultimately, energy was the contractual opportunity here. We're very passionate about new investigators. We invest some of our money into new investigators. We have a leadership organization, which you may hear is called the G8, even though there's not 8 people. We also have sort of an investigator council, which is our key sites, and then we're very passionate about diversity and inclusion. So that's who we are. I'm going to now transition into the title and content of our satellite symposia, but that introduction was critically important to what we're trying to do together. So we have this broken up into an endometrial section and an ovarian section. We're on time. I want to create a little bit of a backdrop for the endometrial section. I'll say a few words, which I'm doing now, obviously, and then we'll talk about trials in progress in endometrial cancer. We'll have a discussion, and then we'll do the same content, introductory content, trials in progress, and discussion for ovarian. I think all of us know that endometrial cancer has exceeded ovarian cancer in its annual estimated deaths, with being ranked 15th incident cases and 19th in the most lethal. And you may say, well, that's good, because it's 15 and 19. It's not good. And we have some opportunity here to do better with 420,000 new cases and 97 deaths. It's a global disease. This is a heat map. You can download this for any cancer in any situation. You can see the darker areas, such as my country, such as Australia and New Zealand, where and some of the European countries where this is a bigger problem, but no matter where you live, it is still a problem. We made a lot of progress all the way from progesterone in 1961, the year I was born, to staging, 1988, the year I graduated from medical school, and ultimately to the TCGA and immunotherapy. This immunotherapy train has really launched now for 7 years, with the single-agent activity of pembrolizumab in 2017, in 2019, pembrolizumab in the mismatch repair proficient, and ultimately now we have multiple options such as dostarlimab. What I'm really most proud of is what these guys have done together. Think of this, with 3, and it's not just the FDA, but 3 FDA approvals in endometrial cancer this year alone. Think of that. So if you can get an FDA approval or an EMA approval and so on, you can change the world, and we've done that 3 times in endometrial cancer this year with an all-comer from GY018 and ultimately on Ruby and all-comer on the right, and then in the middle, DuoE in the DMMR situation. So this is really sensational. We're moving on. We're moving on looking at P53, though. P53, if you didn't realize it, mutation is an activity indicator for bevacizumab. We have a energy trial which is launching, led by Amanda Nicholas-Fader, to look at P53 as a biomarker for bevacizumab in the PMMR subset compared to checkpoint, and maybe we should use all 4 drugs: carboplatin, paclitaxel, bevacizumab, and pembrolizumab in P53 mutated. And then in the we'll hear about selinexor as a great opportunity. We live in an ADC world. We have several ADCs in the clinic. As you know, on April 5th, we got accelerated approval for trastuzumab deruxtecan, but we're moving on, looking at other folate receptor alpha, TROP2, B7H4, just to name some. These DNA damage repair molecules, generally small molecules, generally oral, have been amazing. WEE1 inhibitors, which you're familiar with, adavosertib and azenosertib, and certainly PARP inhibitors are in that class. And then finally, hormonal therapies. We've done a lot of good work. You've done a lot of good work of studying, CDK4/6 and anti-hormonal therapies, and I think this continues to be a high unmet medical need. We're trying to move checkpoints into the front line, C93, pembrolizumab, Dominica dostarlimab. We tried in the PMMR subset with LEAP, did not meet its primary endpoint of pembrolizumab in the PMMR subset. So that's the context. Now we get to hear from these guys, okay, about what trials are ongoing today. We're going to begin with a talk of Professor Colombo and Fujiwara, and I'll turn it over to you. Thank you.
Okay. Thank you, Brad. Yes, I would like to talk about TROP2. TROP2 is an ideal target for ADCs, for antibody-drug conjugates. What is TROP2? TROP2 stands for trophoblastic cell surface antigen 2, and is a transmembrane glycoprotein, very important. It's a cell adhesion molecule, but very important to regulate proliferation of the tumor, migration, invasion. And as you can see, TROP2 is actually overexpressed in several solid tumors, and it is expressed in endometrial cancer. And as you can see, it is expressed across different histologies, even though the majority of endometrioid cancer will express TROP2. But also it's very important to see that there is no relationship according to the microsatellite status, for instance. So the expression is high in the MSS tumor as well as the MSI tumors and in carcinosarcoma. So this could be really an ideal target for ADC and for treatment in endometrial cancer. And the drug I want to talk about is sacituzumab govitecan, which is a TROP2 directed ADCs. As you can see, there is a humanized monoclonal antibody which selectively binds to TROP2 with nanomolar affinity and is internalized. And then the cytotoxic payload, the SN38, which is an active metabolite of irinotecan, so a top of one inhibitor. And of course the linker is a hydrolyzable linker, and so this is important to confer a unique drug release profile, including a bystander effect. And here is the trial that we are running. This is a collaboration between GOG and ENGOT, and is a randomized, open-label, phase 3 study of sacituzumab govitecan versus treatment of physician choice in participants with endometrial cancer who have received prior platinum-based chemotherapy and anti-PD-1, PD-L1 immunotherapy. So you see here the key eligibility criteria. These are patients with endometrial cancer up to 3 prior lines of therapy, including immunotherapy, IO. and of course radiologically evaluable disease and good performance status, and they are randomized to receive sacituzumab or treatment of physician choice, being either doxorubicin or weekly paclitaxel. The treatment will be until progression or unacceptable toxicity, and we have two co-primary endpoints. One is PFS by BICR and OS, so both PFS and OS are primary endpoints. And of course, secondary endpoints are overall response rate, duration of response, and so on. So as I said, this is a trial in partnership with GOG and is actually now currently recruiting. They started in the US, hopefully we will start soon in Europe. And so looking forward to the participation of many of you. Thank you.
Thank you, very excited about that.
So moving on to the next TROP2 drugs, as Dr. Colombo said, that the endometrial cancer has a high expression of TROP2. So this is the MK27, known as a sacituzumab tirmotecan. And this is a randomized phase 3 trials targeting recurrence of the endometrial cancer or carcinosarcoma less than 20 months after the completing the platinum-based adjuvant therapy, or received the platinum in the metastatic settings, or received an IO-based regimen in the recurrence setting after initial adjuvant platinum therapy, regardless of platinum-free interval. So those patients must receive the platinum-based chemo plus IO. So those patients will be randomized either a single agent of MK27 or treatment of a physician's choice. So this trial has a dual primary endpoint, which is PFS and OS. And the secondary endpoints included the OR, or duration of the response, and the stratification factor, as you can see, the MMR status and the TROP2 status.
Thank you, excellent. That's great, thank you. You know, it's interesting, this is really a global effort. You say, well, why do we need all these people? Because we have two trials with very similar mechanisms of action, in fact, the same antibody, right? But different linker payloads, govitecan and tirumitecan. And so I love this slide, thank you. This is a great slide which shows the important collaboration. But let's keep rolling. Let's talk about a third trial with another exciting agent with a very unique mechanism of action. Thank you.
Yeah, thank you. So this is moving beyond the ADC, and here really being biomarker driven. And here is in the box of TP53 wild type. And I think maybe you have seen the update on the SIENDO trial that was presented at the meeting here, really showing the long follow-up of the SIENDO trial in the subgroup analysis of P53 wild type, showing the benefit of the maintenance of the selinexor. But it was not powered to answer the question. So now we have this phase 3 randomized trial really trying to answer this specific question, targeting the patient endometrial cancer, P53 wild type by NGS, and is targeting the patient with a primary stage 4 or first-line recurrent endometrial cancer. The patient will be randomized after platinum-based chemotherapy with response, partial or complete response, and they'll be randomized between selinexor maintenance versus a placebo. And the duration as part of this trial is 3 years, unless there is toxicity or progression prior. And the primary endpoint would be the PFS by investigator assessment, with a secondary endpoint to be DOS. So that will be an important trial to answer the question following the SIENDO update that we heard today, yesterday at the meeting. Thank you.
Thank you, very exciting. Again, lots of discovery in endometrial cancer. I couldn't be happier. Now we're going to transition to another ADC target. Thank you, Professors Fujiwara and Colombo. So let's hear about this ADC.
Yeah, I am happy to introduce a coming study sponsored by BioNTech. I'm now going to introduce the BNT323 study. The investigational drug is called TPAM, which stands for transmab pamilitacan. It is a HER2 ADC, and its mechanism of action is similar to that of transmab dextrocan. But it differs in its toxin and proprietary payload. The phase 1-2 trial encompasses comprehensive data on endometrial cancer with 145 patients, including immunohistochemistry 1, 2, and 3 positive. The breakthrough designation based on this robust data will be presented at the upcoming Congress, maybe next year. I have reviewed the data under a confidentiality agreement, and the results look promising. Yeah, I present an overview of an exciting international phase 3 trial comparing TPAM with the investigator's choice of chemotherapy for patients with HER2-expressing recurrent endometrial cancer. This is the first phase 3 randomized trial in HER2-expression ADC study in endometrial cancer. This randomized multi-site open-label trial aims to enroll 420 patients. The patients will be randomized 2-to-1. 280 will receive TPAM monotherapy, and 140 will receive the investigator's choice of chemotherapy, such as doxorubicin or paclitaxel. Key inclusion criteria include recurrent metastatic endometrial cancer with HER2 expression, levels of 1, 2, or 3 positive, confirmed by central testing, and at least one prior line of platinum-based therapy and one immune checkpoint inhibitor with a maximum of 3 lines of prior therapy, excluding endocrine therapies. Measurable disease for RECIST version 1.1 and an ECOG performance status 0/1. The primary endpoint is progression-free survival assessed by a blinded independent central review. In contrast, a secondary endpoint include overall survival, investigator assessed PFS, and overall response rate, and the duration of response and safety. This trial aims to establish the efficacy and the safety of BNT323 DB1303 in this patient population. Thank you.
Thank you. Thank you. Thank you. So that's 4 trials. You guys got some work to do. That's over 2,000 patients, right? So I think that's important to realize, is that that's why it takes, as some people call it, a village, right? We have lots to do. So I'm going to call her Graziella. Grazi, she's my friend. They're all my friends. So Grazi, tell us why it's important to work with LCOG. So I get the sense as I talk to sponsors, they're like, what do I need those guys anyways? We can do trials in Brazil and Argentina and all of these wonderful places without you. Can they? And if they can they do it as well as they do when through collaboration? Tell us, we understand what the what is. You told us. Now tell us the why.
The main reason, I think, is we have in Latin America 1.3 million cases of cancer every year. So to have a high increase of referrals, increase of patients in the clinical trials would be so important to increase, to include Latin America and LACOG for all clinical trials. And to increase faster also. Another topic that I think is important when we think about diversity. So when we think about Latin America, it's a mix of cultures, mix of Indians, African Americans, white Caucasian people, European descendants. So we have high diversity of genomic patients, genomic profiling, and this can also be a more real-world data when we increase. I think we're very organized in including patients very fast. So after we started the GOG partnership, we included a lot of patients very fast and in different sites in different countries. So I think it's a great opportunity to increase the Latin American participation in all those trials.
Thank you. And we're getting better, but we're already very good. Nicoletta, Professor Colombo, one of the things I think that NGOT does really well is communicate. So you have a very cohesive group. And respectfully, if you work for a CRO out there, I don't mean to insult you, but I have this thing in my email where I can actually put you into spam. Not that I would ever do that for a CRO email, but facetiously. But tell us about the communication and how you keep your sites together and all pushing and they're pulling in the right direction. The solidarity model of NGOT is really, really fantastic.
Yeah, I think we were quite successful, and we did so many trials such a fast way, and we reached the goal that we wanted. Of course, it needs a lot of solidarity, I think, and organization. So we have a very nice structure where there is one leading group, which is one of the country, and so we assign the trial to this group, and then this group will coordinate all the collaboration across the different angle groups, together with the industry, if it is a sponsored trial, of course. But I want just to remind you that we are doing also a lot of academic trials.
Right.
And actually, you didn't mention ATTEND because, of course, the FDA approval was only for those drugs, but we also run an academic trial in the same disease setting. So I think the cohesion and inside NGOT, we trust each other. Is a lot of trust.
Yes, trust.
A lot of friendship, I must say, because we are all friends, and we respect each other.
Yes.
There is a lot of democracy. We want to keep this democracy. And so far, we have been able to do so. So even the leadership of this trial is equally now distributed according to specific criteria and like a scoring system to be able to get the opportunity to everyone, to be leader in a trial, which is very important to coordinate. And then we have also a very strong operational group. Because in any cooperative group, of course, you have the physician, but then you need a very strong operational organization. Mm-hmm. And also they work together, and so they, exchange their idea and expertise together, and this will increase the possibility to grow and to learn and to become more and more, experienced in this respect.
The other thing that you taught me is that you are the future customers of an approval. Mm-hmm. So if you do the study, when it gets approved, you're familiar with it. I think, Razi, you taught me that in Brazil too, right? So if you did the study in Brazil with cemiplimab, when it gets approved, we already know about it. We did it, right? So it helps from sort of an adoption sort of setting. Kaiichi, in Japan, the drug development and the regulatory, it's very different. Yes. Right? So tell us, what's your FDA's, what's it called again?
PMDA.
Tell us about that.
So the regulation in Japan by the PMDA required the safety data, at least, for the Japanese patients.
Right.
So they required phase 1 trials for the Japanese patients before going into the phase 3 trial. But that is the classical way. So based on these the issues, now the many of the bio companies and the venture company do not want to bring the new drugs to Japan. So we realize that this is a big problem because now we are calling it Japan passing. Okay. Right. So we realize that we have to reverse the situation. So, and the one important lessons that I have learned through the GOG and energy investigator-initiated the registration-directed trial, like a GOG 218 or energy GY 018, just finish the PMDA inspection, and we got the pretty good results from feedback them. So the Japanese regulatory people is much more friendly to the academia.
Right.
And then I have the experience to accompany with the pharma people, but they are very, very, strict. However, we have learned that there is a chance that the academic people can contribute to make the better suggestion
Right.
from the PMDA. Yeah.
And so GOT-J, so why would you work with GOT-J? Because they actually understand it, and because you can message.
Yeah.
So this is a great, now, Korea is a small country but mighty, okay? Korea has been a,
Small.
what?
Not small.
Not small. Mighty, strong. And you enroll very well, and we're grateful for your contribution to many of these studies. Tell us why they should work with the KGOG. Give us an explanation why the KGOG is an important partner in the clinical trial process.
Yeah, KGOG try to get ability to get expertise to conducting clinical trials and getting huge experience about clinical trials. And the second is we get a national network, and number third is we get reliability to clinical trials. So with these portions, we can suggest our KGOG's ability to sponsors
Right.
good clinical trials. So we get monthly meeting with face-to-face meeting or sometimes Zoom meeting. Korea's hospitals, more than 70% are located around the capital of Seoul. So it is very easy for us to meet each other.
Right.
So huge, big trials and small trials can be possible in Korea. So we can explain our KGOG's ability to sponsors, industrial sponsors. It is our desire.
And thank you for hosting a wonderful IGCS last year. It's a wonderful opportunity to visit your beautiful country and enjoy your hospitality. Yeah, right? Thank you. So Canada is an interesting partner, also very good enroller. I find, though, that some of the medicines, let's say mirvetuximab, we're going to talk about ovarian in a minute, are not yet approved there. So tell us why that's important, let's say, in doing drug development in certain scenarios, such as in folate receptor alpha high studies, which we're going to get to soon.
No, it's a great question, and I think it's why it's important to have trial options. So it's why it's very important to be part
Right.
of this network, because depending on the health agency, the approval can take more time. So it's important to ensure that we offer the best treatment strategy for our patients. So having a portfolio of trial allows us to actually try to mind the gap while we wait for
Right.
potential approval. And clinical trial network is also about speed, right? If you
Right.
work through the cooperation group, you can gain time. And I think that is very important, because as we see all the trial coming, it's very important to accrue quickly, because your control arm may change. And it's also why collaboration internationally is important, because you may have different options in different countries. So that helps actually to answer a specific question that remain relevant, but may be different in different countries. So I think it's why this collaboration internationally is very important.
Yeah, thank you. There are two pieces of misinformation that I heard at this meeting. Someone said, we don't really want to work with the GOG because there are other sites, other than GOG sites in the US, that want to do the trials. Who are they? Right? The second is what you just said, speed. Well, we think it's faster to work outside of the Canadian Cooperative Group. No, it's not. So if we can actually engage through communication, solidarity, and friendship faster with this is why we're here. This is why these networks exist. And that's really a great summary, and I thank all of you. So let's now move on to ovarian cancer. Ovarian cancer continues to be a very serious disease. Not quite as many new cases or deaths as uterine, but still very important. 324,000 new cases worldwide, 206 deaths. Very serious. And here you see again the color. I'm sorry, Stephanie, that you're in the darker color here. But I hear you. Italy also is pretty dark here. Thank you, Brazil, for being a little lighter. But this is a global disease and very, very important. I think all of us know that ovarian cancer is unique and that patients get several lines of therapy. And that creates opportunities to impact the life cycle of the disease, the natural history of the disease, whether it be at the first response, platinum-sensitive recurrence, or platinum-resistant disease. One of the unique aspects of epithelial ovarian cancer is the heterogeneity. And we've done a great job in some of the high-grade serous cancers. We're going to hear in a minute about a low-grade serous cancer. We're very interested in these rarer tumors. I just was stopped in the hall about a terrible clear cell cancer case, and we're very interested in these heterogeneity and trying to do trials that are interpretable in homogeneous groups. It's not just the histology, it's the molecular signature. For example, we're doing studies with ARID1A. Obviously, you're familiar with PI3K. We're very interested and are going to launch very soon an anti-PI3K study. And obviously, RAS for some of the MEK-FAK inhibitors. So we're really excited. We already heard about HER2 or B2 and so on. And certainly the CDK1 and 2s are in flight. So we've really done a nice job in impacting frontline. It's not enough. And I'm here to remind you of something that I think is terrible. So in 2020, which was a good year, we got Palawan and Prima approved in the frontline. Isn't that great? We haven't done anything since. Okay? And there are several studies, maybe one of which I presented at ESMO, Athena combo, that didn't work. So we have our work cut out. The good news is we're doing several studies in the front line. We're also doing studies with neoadjuvant chemotherapy, trying to improve anti-angiogenic. I think all of you know that in the HRD test negative subset, PARP inhibitors don't work very well. So maybe there's an opportunity to add to bevacizumab, and particularly in the HRD test negative opportunity. So we have these artificial sort of platinum-resistant definitions. We're moving beyond that, but for today, many of the regulatory opportunities are in platinum-resistant. I really appreciate what our European friends have taught us, is that it's really patients that are eligible for platinum. That's more of a clinical rather than a regulatory endpoint. And this is one of my favorite slides, is it's a spectrum. It's a spectrum. It's not black or white, it's shades of gray. We continue to be proud of Aurelia. Again, prior to Mirasol, this is what we were left with. This was very nice in that weekly paclitaxel bevacizumab in the lower right-hand corner had a 51% response rate, and I think in almost all of the settings, we have Aurelia FDA approved. We already talked about antibody-drug conjugates. We talked about TROP2. We talked about HER2, and there's others. And now I'm ready to get into it. The trial's in progress. So we're going to ask Dr. Dalmullen to kick it off. Thank you.
Thank you again. And as just said, the platinum resistant is still a very important gap in knowledge for ovarian cancer. So we'll now discuss some trials about this. And starting with the glucocorticoid receptor modulation. This is a very important pathway that has been explored, and with relacorilant, that is a selective glucocorticoid receptor modulator. And we have a phase 2 trial that was published by Dr. Nicoleta Colombo last year in JCO that showed in different cohorts, especially in the intermittent use of relacorilant, increase in duration of overall response rate, increase in PFS, and increase in OS. And because of this, we have now the phase 3 trial ongoing. And it's so nice to see that this phase 3 trial enrolled almost all this table: US, Brazil, Canada, South Korea, and Europe. And I still have patients that are in this trial. It's very exciting to see. Rosella is a phase 3 randomized open-label multicenter study of relacorilant relacorilant plus nab-paclitaxel versus nab-paclitaxel monotherapy for patients with recurrent platinum-resistant ovarian cancer. For the inclusion criteria, it was included patients that had a progression less than 6 months after the last dose of platinum, with 1 to 3 prior lines of systemic therapy, and all patients must have received prior bevacizumab. For the stratification factor will be region of the world and prior lines of therapy. And all patients were enrolled, 381 patients, to relacorilant plus nab-paclitaxel versus nab-paclitaxel intermittent way. And the primary endpoint of the study is progression-free survival by RECIST per blinded independent central review. And the key secondary endpoints: overall survival, safety, PRO, and quality of life. The last patient was enrolled this year in April, and we're very excited to wait for the results, waiting until the end of this year. Maybe Dr. Colombo will say something, who knows? And moving now again to the ADC era, we also have another ADC for ovarian cancer being explored, is the reladatatul deruxtecan, or RDXD. We have a phase 1 trial with this drug that was presented by Dr. Kathleen Moore in last ESMO. And we saw very interesting safety and response rates of 40%. So that's why now we have this phase 2 and 3 trial that is called Rejoice Ovarian 01. So for this study, all the key inclusion criteria are high-grade serous or endometrial ovarian cancer with 1 to 3 prior lines of therapy and needs to include bevacizumab, platinum-resistant. Patients should have received MIRV if have a high folate receptor alpha, and of course if MIRV is available in the region, ECOS 0 to 1, no prior CDH6 target inhibitor, and all patients must be can be excluded platinum refractory disease. RDXD is a CDH6 antibody against antibody that is a cadherin 6 cell adhesion protein. This trial will have 2 parts. The phase 1 to find the optimal dose, and patients will be randomized for 3 different doses, 4.8, 5.6, or 6.4, and then follow until progression of disease. The primary endpoint, overall response, blind independent central review, and secondary endpoints, overall response per investigators and duration of response. This phase 2 is already enrolling some sites. And later, with the optimized dose finding, we'll have the phase 3 part that will randomize for RDXD or treatment of physician's choice, including gemcitabine, pegylated liposomal doxorubicin, topotecan, or paclitaxel, until progression of disease. Primary endpoint, overall response rate, PFS, and secondary endpoints, overall survival and quality of life.
That's great. Thank you so much. That is so exciting. About Rosella. She was very passionate about presenting that. Even though that study has completed enrollment, that should be the next platinum-resistant recurrent ovarian cancer study to give results. So that gets me fired up. And then I also liked what you did, CDH6. Oh, we got another ADC target, right? So mirvetuximab soravtansine, the Mirasol study, which you're very familiar with, has been very successful. It's a medication published in the New England Journal, and you're right, it's going to be available in several parts of the world. Folate receptor alpha is a good target, but maybe we can do better.
And because we don't have mirvetuximab yet in Canada, we need to have other options if folate receptor is a good target. So what can we do? So I'm presenting this phase 2/3 open-label study evaluating the efficacy and safety of Luvelta in patients with platinum-resistant ovarian cancer. So as most of the ADC, there's often now, as part of clinical trial design, a dose optimization finding to ensure that we hit the efficacy without significant safety issue. So this trial actually has been built with a part 1 to define what is the appropriate dose to use for the randomization part. So you have cohort A with a specific dose and a cohort B with another dose, and that has been completed accrual and is expected to have the dose finding available in December. While we wait for this dose finding, there's currently the part 2 ongoing, randomizing patient versus cohort, like the dose of A, dose of B versus standard of care, and the standard of care chemotherapy is a usual standard of care with gemcitabine, weekly paclitaxel, topotecan. And then when we will have the right dose to use for the randomization will be 1-for-1 with a dose that is used and optimized versus the standard of care chemotherapy. What is also important as well to say that the main objective is the PFS, but the OS is also an important endpoint that will be collected as part of the trial. And as we mentioned, because currently we don't have necessarily folate receptor mirvetuximab available across the world, it could be an option for a patient to have a specific ADC targeting this antigen. But as you know, I love biomarker, so let me talk a little bit more about the biomarker. So, and just because here, this specific trial will target the folate receptor alpha. This is based on, as a pre-screening for this trial, on folate receptor expression. So you need to be positive. But what does it mean to be positive? A biomarker doesn't mean anything if it's not well defined. So for mirvetuximab, and we know with the different trial that has been done, there's a specific definition looking at the intensity and the percentage of expression. So here, as part of this trial, actually the percentage of biomarker positive will be much higher and targeting 8 over 10 women positive for folate receptor because it has a very different definition. The screening would be based on IHC as well. But the selection is only based on the percentage of staining, which is at least 25%, regardless of the intensity. So that is very different of how we select for mirvetuximab. So we'll actually be able to enroll much more patients than the one eligible for mirvetuximab. And as part of this trial, what has been incorporated, because sometimes when you have patients with platinum resistant, you need to treat them immediately because there's a lot of symptoms. So as part of the trial as well, there's a pre-screening allowed to actually screen your patient for the folate receptor, knowing that the percentage of biomarker positive will be actually 80% of your patient for this specific trial. So I think that is a good option for patients that have this folate receptor, which is 80% of the population. So
That's great. Thank you so much. Luveltamab, tazevibulin. It just rolls off the tongue. So I appreciate that. But there's great opportunity with the evolution of the science of the novel linker payload with an established target of folate receptor alpha. So that's great. And in fact, there's another folate receptor alpha medication that we're excited about. In fact, I refer you, which we don't have time to review in detail, at ESMO, the dose optimization of 100 versus 120 and how it sort of very intriguing preliminary data. So Taichi, tell us about RENA-S.
So RENA-S is RENA-S stands for Rena Tabat Sustecan. It's really hard, so I would say
rolls off the tongue.
Okay, and this is another ADC having a folate receptor alpha antibody plus the exatecan, which is the analog of the camptothecin. So it's very unique. Combination. And as Brad said, in the ESMO last month, showed the very, very good efficacy and the tolerability of the RENA-S in the ovarian cancer patients. Okay, so now it's moving to the phase 3 trials, which targeting the platinum-resistant ovarian cancer patients, randomized to the single agent RENA-S versus investigator's choice of the chemotherapies. So this, in this patient population, you must have the platinum-based therapy, and you have to have the bevacizumab therapy, and the patient also must have the PARP inhibitor therapy, and also in the area in the world, probably in the United States or somewhere in Europe, the MIRV must be used in case the positive of the folate receptor alpha the expression. So this is very, very unique trials showing that this Rina-S may be effective regardless of having prior MIRV2. Okay?
That's great. Thank you, Kaiichi. So, guys, that's more than 1,000 patients with platinum-resistant recurrent ovarian cancer, right? So we got some work to do, and that's why we're here together as a team. So it's my great pleasure to talk about, I think, I could be mistaken, the only randomized phase 3 registration trial in a rare tumor, okay? In a rare tumor. So that's a big effort, and I appreciate you, Jae-hoon and Kim, for giving us your perspective.
Yeah, we KJG experienced, we needed 2 years to prepare and to discuss with this protocol with sponsor Verastem. You can see the schema, and I will introduce RAMP 301 for low-grade serous ovarian cancer. In this morning and yesterday, we found a very good result. RAMP 201 and the phase 2 study it was. There are currently no FDA-approved treatments for patients with low-grade serous ovarian cancer. Abatumtinib targets both RAF and MEK, providing a vertical blockade of the RAS/MEK kinase pathways. Defactinib is a selective FAK inhibitor that mediates adaptive tumor resistance through the activation of the RAS and the MEK kinase pathway. The combination of defactinib with abatumtinib can induce better anti-tumor efficacy with synergism than either agent alone. This study is a schema to introduce RAMP 301 phase 3 randomized open-label international study. It is evaluating the combination of abatumtinib plus defactinib versus the investigator's choice of treatment in patients with recurrent low-grade serous ovarian cancer. Now let's look at the key eligibility criteria for this study. Patients must have a confirmed diagnosis of low-grade serous ovarian cancer, recurrent disease after prior platinum-based therapy, and the documented KRAS mutation status. Additionally, they must have measurable disease based on resistor version 1.1 criteria. And prior MEK inhibitor or bevacizumab permitted. The study randomized the patients 1-to-1 into 2 groups with a total sample size of approximately 270 patients. In the experimental arm, patients receive abatumtinib at 3.3 milligram orally twice a week, along with defactinib at 200 milligram taken twice daily. This regimen follows a 3-week on, 1-week off schedule. The control arm is the investigator's choice of treatment, doxil, paclitaxel, letrozole, or anastrozole, with 135 patients assigned to each arm. Patients are stratified based on 3 key factors: KRAS mutation status, the number of prior therapies, and the geography. The primary endpoint of the study is the progression-free survival, assessed per RECIST version 1.1, and evaluated by blinded independent central review. The secondary endpoint includes overall survival, PFS, as assessed by the investigator, objective response rate, duration of response, disease control rate, safety, and pharmacokinetics, and patient-reported outcome. About analysis, they do interim analysis at 50% of planned PFS events for possible sample size adjustment to maintain power hierarchical evaluation of primary PFS endpoint against KRAS mutant low-grade serous ovarian cancer only and all recurrent low-grade serous ovarian cancer. And it includes a crossover feature on BICR confirmed PD. Thank you.
Thank you. It's excellent. So we've already talked about how mirvetuximab soravtansine has changed the world. We've already talked about how we're trying to evolve folate receptor alpha as an established target. What we do, though, is we get medications approved in later lines of therapy, and then we move them earlier. And then we study combinations and maybe even study other tumor types. So this is really an exciting trial that we're going to hear about here from Professor Colombo, again, earlier and in combination. Thank you.
Yes, we go back to mirvetuximab, and this is one of the first ADC, folate receptor alpha ADC, which is, as you know, approved in many parts of the world in the platinum-resistant population. But now, what is the role of mirvetuximab in the so-called platinum-sensitive recurrent disease? Which is not correct. We don't want to use any more platinum-sensitive. We prefer to say patients for whom platinum is the best option or is not the best option. In this case, is the best option. And so this is the Gloriosa trial, is the GOG 3078 ENGOT OVIS 76. Is this is a trial for patients with platinum-sensitive recurrent ovarian cancer, receiving a triplet therapy, so a platinum-based combination together with bevacizumab. So these patients, at the end of the therapy, of the triplet therapy, they are tested for folate receptor alpha, and if they are in complete, partial, or stable disease, they will be randomized to receive our drug, so mirvetuximab, together with bevacizumab or bevacizumab alone. As you see, the requirements is that patients must receive the triplet therapy, but in case this triplet therapy is not available in that country, there is the option, there are two options actually of enrollment. One for those, this is a run-in option for those where, let's say, bevacizumab is not available in combination with chemotherapy. And the other, which is the main option, is for patients receiving the triplet therapy. So as you see, they have to be eligible for this triplet therapy. They have to be tested for folate receptor alpha. They have to achieve a complete, partial, or stable disease, and then they can be randomized to receive mirvetuximab with Bev or Bev alone. So this is a trial ongoing and rolling very well. I think it's a very important concept. We want to test first this population, because then eventually you may want to bring this drug even more upfront in the frontline setting. And so that's the way we do. I mean, we start with platinum resistant, then we move to platinum sensitive, and hopefully, eventually, we will reach also, and actually, this will be reached very soon, even the first line setting. So this is the Gloriosa trial, and I think this is concluding our ovarian cancer presentation.
Thank you. That was fantastic. Thank you so much. And you said this is how we do it. I think it was Montel Jordan, right, that said this is how we do it on a Friday, just saying. So that's really exciting. You guys, that was funny. I'm really trying here. So it's interesting that many of these are camptothecin topoisomerase-like medications. I'm going to ask you a hard question in a minute, Stephanie. But Grazi, can we study all of these? We've talked about 4 targets today. HER2, TROP2 twice, folate receptor alpha, 3 medications, and CDH6. Can we do all these? Maybe we only need 1.
I think we will be more and more ovarian cancer like lung cancer, that we have multiple diseases in one with multiple biomarkers. So that's why we need so many partners in collaboration to increase the recruitment of patients. I think the most hard thing to include a patient with platinum resistant or not able to receive platinum is because they get sick so fast.
Right.
So we need to have a lot of sites because only maybe in the screening part of the study, a patient can decrease the performance status and we won't be able to be included in a trial. So including different centers is essential to have all these trials done.
I agree. Nicoletta, you and I have been doing this for a while. No secret. We still have a few good years left, as we say. What percentage of PROC trials over the last 20 years have been positive?
Oh, well, unfortunately, not very many, because, I mean, our standard of care is still the Aurelia. I mean,
Right.
just Mirasol came out recently, but if you think our standard of care is still the Aurelia trial.
Right.
It means many negative trials ongoing. But now, I think things are changing for sure, because these new drugs are so effective.
I hope so.
At least in the phase 1 and preliminary phase 2, you see really response rates which are much higher than, and also the duration of response. So positive.
But what I see is I see sponsors, I don't know if I can, run a study in platinum-resistant recurrent ovarian cancer. We got CDH6 and folate receptor alpha and listen. The chance that all those studies are going to be positive, very low. I hope it's more than what we've done in the last decade. But I think, as you said, Razi, we need to get this done. And now, Stephanie, can we give a topoisomerase after a topoisomerase? In other words, if you get a folate receptor alpha linker payload that's the topoisomerase camptothecin, does that exclude, let's say, another target, let's say a CDH6 with a topoisomerase? I think that's the question, and give us your opinion on that.
And I think it's a question that I cannot answer yet. Hopefully in a few years we will, but I think it's why we need to build the trial trying to answer this question.
Right.
So that will be very important. Now I think the sequence of treatment matter for outcome. But I think what is, with all the ADC in development, it seems to be overwhelming. But I think it's very important to look at the characteristic of ADC, because they have their own cytotoxic, their own linker characteristic, the bystander effect. So it's why there's different criteria for inclusion, because they have all their own property for ADC. And if we can, for now what we know from the other disease site, maybe the same payload may not be efficient, but again, I think it will depend on the mechanism of resistance.
Right.
So maybe changing the antigen may have an effect, but I don't have the answer yet, and I think it's something we definitely need to assess.
Thank you. And Jaehoon, you and the KGOG, you're very good at investigator-initiated trials. So tell us about your investigator-initiated trial process. I noticed that you do several of those. Tell us how that works, because not every trial needs to be sponsored by a drug company at the level of regulatory approval.
You mean KGOG's experience about IIT?
Yeah.
We have frankly said that sometimes we receive without money study, just IIT from the scholars.
Yes.
And some protocols with just drugs without money.
Uh-huh.
Yeah, sometimes very rarely drugs and study money. So we should get a monthly meeting and discuss. We determine the order supply, which one is first and the second. Yeah. So it is very difficult. So GOGP and ENGOT should supply to KGOG.
Try to do more.
Easy study.
The last question, I'm going to open it up to you, so maybe you can think. We'll have just maybe 5 minutes for questions from the audience. We have not included advocacy groups in this conversation. And I apologize twice. If you have a study and we didn't discuss it, I'm sorry. And many of the studies we talked about were not sponsors of this, so you didn't have to be a sponsor for us to discuss your trial. That's important. But Kaiichi, under your leadership, the advocacy has been really, really important. Tell us how we can expand that role in recruiting, in enhancing enrollment through advocacy, and I think the IGCS may be one of the main leaders in that. And you're very passionate about that, so tell us about the advocacy initiative in IGCS.
Well, thank you for asking this. Yeah, we have the close communication of the patient advocacy group for about 10 years now. And the most beneficial things for both of us is that we can exchange what they need
Right.
and what we need. And sometimes patients can help us. For example, the rare tumors. The patients with the rare tumors having a problem and they are looking for something new. So that is one of the most important key factors to communicate with the patient's advocacy.
Well, thank you for your leadership. Are there questions? We have some microphones here. Are there any questions from the group? This has been a wonderful conversation, and maybe I don't see any questions, so maybe we'll bring this to a close. This concludes our symposium. I really want to thank our presenters and to you, our audience, for joining us this afternoon. The IGCS and the GOG Foundation would like to thank our commercial supporters for this independent medical education: Gilead, Karyopharm Therapeutics, Genmab, Immunogen, Corcept Therapeutics, and Sutro Bio. And if you would like to help us, we need your help. If you work for a sponsor, we would appreciate it if you would contact Jenna Cummins, who's here in the front row, who would be more than happy to work up a grant for that opportunity. So we hope that this is a sustainable opportunity. We hope to see you in South Africa, and thank you for what our group said. Did you have a final comment?
Yep.
Okay, go ahead.
My final comment is the appreciation to the GOG Foundation for supporting this fantastic symposium. So,
thank you, El
really
Presidente. All right, well, thank you, and enjoy the rest of the meeting. Thank you.